Thank you very much, everyone, for being here and for inviting me. I want to introduce my co-workers, my wonderful colleagues that this project would not be possible for without them. Dr. Valerie Copie, who's a biochemist at my university at Montana State, and she's analyzing the metabolism of FD patients and families, and we're doing that in patients and in mice. And my other esteemed colleague, Dr. Seth, who's a gut microbial ecologist and is really an expert in understanding all the bacteria in the gut and which are healthy bacteria, which are not so healthy. And we really want to compile this information to ultimately come up with treatments for FD is the ultimate goal. But the need was to just at least understand what's going on in the FD gut. And we know there are a lot of gut issues in FD, and so we wanted to start with a big picture view and figure out what's going on with the gut microbes, what's going on with the nervous system in the gut, and how do these things together affect the metabolism, meaning what kinds of chemicals patients absorb or don't absorb, how that affects energy levels in patients. And so we got a nice grant from the NIH to do this in both mice and in FD patients, and of course none of this would be possible without Lucy and Horacio and Alberto in the Dysautonomia Center, and of course all of you who are providing samples for us. So it really takes all of us to accomplish this. And so that's who we are and what we're doing. And I'll just say one last thing. My lab has been studying FD for about 10 years. I also have two cousins who had FD, so this is very meaningful to me. And there are some other approaches we're also taking. We're interested in the retina and FD, but that's a separate project that I won't be talking about today. So what you might not realize, and it blew my mind, I certainly didn't know this, is that your gut has about 100 million nerve cells in it, okay? And I don't just mean nerves coming into the gut, I mean the actual cell bodies, the cells themselves, sit in your gut, okay? So there's about 100 million of them, and they're there in green, and they're innervated. They get input from all kinds of other neurons that come from outside the gut, okay? So in red, you're seeing axons that are coming in from outside the gut, and in green are the neurons in your gut. And these neurons from outside your gut are coming from the brain stem, from the spinal cord, and from regions in your neck and regions outside of your spinal cord. And so we know in FD there's a problem with the nervous system, and so what we are hypothesizing is that that problem with the nervous system may be exacerbated or improved depending on the microbes in your gut and the entire metabolism. So here's just showing some of these interactions. You have a brain. You have a brain stem. This is showing the gut. The gut's full of bacteria. It turns out the bacteria actually make chemicals that activate neurons. So the bacteria actually make neurotransmitters, for those of you who know about neurotransmitters. And so there's real signaling going on between the bacteria, the cells that line the gut, and the neurons in the gut, and the neurons that innervate the gut. So this is showing some of this innervation. And so on the bottom, you're seeing the gut, you're seeing the neurons inside the gut, and you're seeing nerves coming in from various regions outside the gut. And we know that many of those populations of neurons outside the gut are impaired in FD. And now we want to take a closer look at how the impairment in those nerves might be altering the gut to cause some of the gastric problems that many patients, FD patients suffer from. So I'm going to turn this over to my colleague Seth.
So I'm a microbiologist and I speak about microbes a lot, but a lot of folks that aren't trained in microbiology sort of have a problem because we can't see them. And because we can't see them, we don't think they're real, or we connotate things like all bugs are bad, all bacteria are bad. We only know about them because they make us sick or they infect us. So I just wanted to give some visuals that might help us understand the size, the scale of microbes. So on this blue structure, you can see in yellow, these are individual bacterial cells. And you might not know what that blue structure is, but if I did this, it's actually the tip of a needle, a sewing needle. Okay, so this is the scale that we're talking about. And you can see that there's thousands of bacterial cells just on that little tip. Now, when we're talking about the gut microbes, these organisms that live in the GI tract, these cells just aren't inanimate. They take in nutrients from our diet, and they process that, and they release other molecules based on their metabolism, and then our cells can get those metabolites or those compounds, and oftentimes they're beneficial. They give us vitamins, they give us metabolites that boost our immune systems, they help make the gut make a barrier against invading pathogens, and so they do many wondrous things. And the surface of our gut, shown up here, you can see all these microbes that are living right on top of our cells. So this is normal, this is healthy, this is not pathogenic, this is not deleterious, this is things that we need, but yet things that we don't know about. We don't really understand what compounds are made, what types of bacteria are there, but what's the potential for human health? Well, here's the GI tract, and you can imagine if you stretch this tube that goes throughout you out on the floor, which would be kind of gross, but if you did that, it would be on average about 23 feet long and about the size of a garden hose, right? About 1.5 inches in diameter. Now, you can, if you remember some high school geometry, you can figure out how much surface area that would be just simply by laying it out, and that would come out to be about 9 square feet. But I'm here to tell you, and you might not realize this, that the actual surface area of your GI tract is about 350 square feet. So if you ever lived in a small one-bedroom apartment, and I know if you're in New York, you probably lived in some place about 350 square feet, because my hotel room was probably around that last night. But that is a big area for microbes, okay? And the reason that you can go from a theoretical 9 square feet to 350 is because of the architecture of the GI tract. There's finger-like projections that we call villi, and if you zoom in on those finger-like projections, along those projections are comprised of epithelial cells, and you zoom in on those and you see these microvilli. Okay, so you have villi that look like invaginations into the GI tract, and if you drill on those, they have hair-like projections called microvilli, and this effectively just makes the surface area huge. And so when we talk about the microbes, we can go from sort of the tip of a needle, and ultimately what we end up with is the size of about a badminton court. And it's a terrible analogy because hardly anybody plays badminton, so I put in a picture here, but it's a huge surface area. So you think about the numbers of bacterial cells that could potentially colonize this area. This is potentially a huge factor of our health and disease that we're trying to figure out and understand. And as Francis told you, all of this surface area is innervated. These interactions between our cells and between nerves with microbes is happening constantly, and it's really a black box, and we're here to kind of figure out how this is happening. This is sort of the overview slide again, just emphasizing that we're talking about both sides of the fence here. We're talking about nerve cells up here, bacteria down here, and the go-between being these metabolic compounds that we think can help promote neuronal health, slow down neurodegeneration, and potentially be very important to help mitigate some of the symptoms in the GI tract that a lot of folks that we've heard about today just by talking with folks and families. So this is the area of our study. We'd be happy to take any questions that you might have.
Thank you.
Yeah, thank you.